if you randomly lose people, that could have an impact on the study. And again, there's a loss of power and there's other forms of bias. So let me just show you this kind of cooked up example. This is from the fracture intervention trial, which was a trial of alendronate versus placebo. And one of the outcomes was vertebral fractures. And in order to assess those, we had a baseline X-ray and then an X-ray at the end of the study, a vertebral radiograph, and those two were compared by an expert reader. So we had the baseline, we had 2 years, and we had 3 years. And a vertebral fracture was defined by changes in the vertebra over the course of the study. Now, we were really good because we did the study, and we actually had follow-up radiographs on 97% of participants at the third-year visit. So remember, we had a 2-year follow-up and a 3-year follow-up. And if we looked from the baseline to 2-year visit, we found the relative risk was about 0.34. Baseline to 3-year, it was about 0.49. So the treatment effect varied over time. The treatment seemed to be kind of doing better in the beginning than in the end. But because we had 97% of people at 3 years, we could kind of do this little thought experiment, which I call a virtual experiment. And so we said, well, we had follow-up X-rays on 97% of the surviving people at year 3. What if we had less complete follow-up? What if we had randomly lost half of the people between years 2 and 3? How would that change results? So if you do that, so here's the original results I already showed you. The relative risk is 0.34 at 2 years, 0.49 at 3 years. So if we then say we had the 2-year X-ray plus 50% of the 3-year X-rays, the relative risk goes down to 0.37. You can see what's happening here is because you've lost some of the 3-year, that the 2-year effect kind of begins to dominate. So even in this case of totally random loss to follow-up, you can see that there's an effect on the outcome of the study. So in the usual case where you have actually some bias in your loss to follow-up, that can really affect the study as well. Okay. So this is kind of a cooked-up example in the sense that this is a study where the early results varied from the later results, so it created bias even from within the study, and therefore the random loss to follow-up even affected results. But again, most losses to follow-up are not unbiased, so there's more reason for concern here. Okay, before I go on, actually, I want to take a little break, and I brought my little compliance roadshow here. Now, I used to talk about this a little bit more, but I know in this class now we tend to focus more on interpreting studies than actually doing studies. But for those of us who have done studies, we think, well, what can you do? And there are all these little things you say, this sounds like a silly little thing, but I think it really helps people feel like a part of the study. So, for example, this is from the FIT study. We made up these little calendars that we gave out to people. It says FIT study, has the advantage that the participants can write their visit dates in the calendar. This is an example of a brochure from the HERS study, and I'll just leave this stuff up here. You guys can come look at it after the lecture. And then one of the things that we've done a lot of, and a lot of people do now, is we actually create newsletters. This is called As You See Fit. So we created a newsletter and sent that out to all the participants. And the newsletter had things a little bit about the study, but about osteoporosis. I mean, there was sort of gossip about some of the other participants. Now I don't know if it would be HIPAA compliant, but things about that. For example, there's a study here about DHEA, and we talked about the importance of calcium, and there were high calcium dietary recommendations, and various things. But the idea here is, again, you're trying to make the people in your study feel like they're part of the study. This is from a study called the Study of Osteoporotic Fractures, which is a cohort study. It was done. One of the sites is in a place called the Monongahela Valley, the Mon Valley outside Pittsburgh. They had about 2,000 participants, and they made up this very sweet book that had like little pictures and stories about the participants that they distributed to each of them. And the idea was to kind of make them feel, again, a part of a larger whole, to really get them, to educate them and to kind of create a sense of community. And then they also, Pittsburgh, that group is particularly good about this. They also made up a little cookbook, favorite recipes of the study of osteoporotic fractures. So I mean, it depends on how big a study you're doing and everything. You can do all this stuff. And we gave these bags fit bags, which I still have, which I remember cost about 90 cents to produce in bulk, so very cheap. But the nice thing about the bags is people like them, but also people were required to bring in their medications to each visit, all the other medications. So we use the bags for them. So any of you doing trials, I know at least there's one person in the audience doing a trial, you might think about these kinds of things to kind of increase people's sort of commitment to the study. One of the other things that it was interesting in these large cohort studies, but it could also happen in a large clinical trial. They've done things like lunches, like had participants come and invite all the participants to come to a lunch, which has a kind of a nice advantage of allowing people to meet each other and creating, again, a sense of community. And also people have done things where if you transport people in a van, the participants get to know each other. So there's all kinds of things you could think about. Okay, so this is now, I'm going to sort of talk about the impact of missing data on the primary outcome. This is from the Hollis and Campbell article in BMJ, and I think this is a really good article. It was in the required reading. And if you hadn't read it, at least look through it, because I think they have a lot of good things to say about missing data and compliance. So let's see, is this it? No. Oh, so this is just, what they did is they looked at the different sort of outcomes they could have. And in this case, let's see, this is a trial of different ways of handling missing data. And the outcome here was balloon angioplasty versus stent. And they were looking at restenosis rate. That was the outcome. And so here is restenosis at 6 months was the main outcome. The answer was yes in roughly a third of the people, no in a little more than a third, but unknown in a fairly high proportion. And so they looked at the results in different ways. So if you looked at the complete case analysis, that would just be, I guess, these two groups. Just the complete data, they got a absolute difference of 9. If you assume poor outcomes, so you assume that in every one of these cases there was unknown, they had poor outcome, you can see the results are quite different. See, I guess that's right, yeah, 67 here, so that's 30 plus 37. If they assume good outcome, you can see it's quite different. And then they can talk about extreme cases favoring stenting versus angioplasty. So depending on what you assume about the missing data, it can be quite different effects. So it's not a trivial problem. Okay, and again, I've really already talked about this. The impact of the lost follow-up is the groups may no longer be comparable. And again, you lose the advantage of randomization because the groups, if they're lost in some biased way, are no longer going to be comparable. Okay, so one thing that people like to do in trials is measure adherence. It's really helpful to know, first of all, during the trial, if you find a patient is not adherent, maybe you can counsel them. They say, why are you not being adherent? And they said, well, it's kind of a pain. I thought I had to take my pill every Thursday, and Thursday is my bingo morning. And you say, no, you can take the pill on Sunday, whatever. So I mean, it's really good to measure it. And I think when you get all done with the trial, particularly if you have a negative trial, you might want to know what was the adherence, and also you might want to do, we'll talk about in a few minutes, a per protocol analysis. So how can you measure adherence? Well, there are whole articles on this, but one thing is you can just ask people, and that's the most common thing that people do, and it's actually been validated over and over as a really good thing to do. So you can do self-report, and that can be where you ask people when they come in the clinic, in the last 7 days, how many of your pills have you taken? Oftentimes, people are given a diary. They write down every day, and then they collect those diaries in the clinic and assess compliance. I mean, of course, it relies on people telling the truth. But again, most people will, so that's pretty good. You can do pill counts. That's, again, often done. You ask people to bring their unused pills to the visit, and then you count the pills, and you can figure out how many they should have taken. For some studies, depending on what the thing you're measuring, you could have a biochemical assessment. I think it was a selenium study that we talked about last week. I think they did some kind of measurement of blood selenium levels, which could tell them if the people were taking it. And then there are these high-tech pill bottles, which I'll show you a picture of in a minute, that actually record every time the patient opens the pill bottle. None of these things are perfect. I mean, they're all problems. Now, if you're talking about adherence in terms of the visit schedule, there, in terms of metrics, you could look at just the number of missed visits.